This video is B6 in a nutshell. It's everything that a double student, that's a combined science student, needs to know for the topic B6, and it can be boosted up with the triple science content for triple science students. So the first thing that we're going to look at is sexual and asexual reproduction. Now, the definition we must use for sexual reproduction is the fusion of gametes. Okay, the gametes could be egg and sperm if we're looking at animals. It could be the pollen and the ovule if we're looking at plants. Okay, so flowering plants use sexual reproduction, and it's the fusion of gametes. That is our definition. It provides genetic variation because there are there's genetic information coming from two separate individuals. And then we've got asexual reproduction. This is where genetic information all comes from one individual. There is no fusion of gametes. Just down here we can see a spider plant. Spider plants are non-flowering plants, and you can see they grow these tubers that have a small spider plant at the end of them. And if that was in the forest, that small plant would land on the floor, start to grow some roots, and it's a really good way to quickly colonize an area. Okay, no fusion of gametes. So sexual reproduction, fusion of gametes, gametes being the egg and the sperm. Asexual reproduction, no fusion of gametes, all genetic information coming from one individual. Meiosis, one of our 3 words in paper 2 that we must spell correctly. It's all about me-osis. Okay, some people say meiosis, I say meiosis. It doesn't really matter, but we must get our spelling exactly right for meiosis. So even though mitosis was in paper 1, we may see a comparison, maybe a comparison table used for mitosis and meiosis. So mitosis, just recap from paper 1, is all about growth and repair of cells. Okay, it happens everywhere in the body except for the reproductive organs. There's only one division involved, and it creates all cells apart from the gametes. Now, because those cells produced in mitosis are identical, the number of chromosomes, if it were a human cell, is 23 pairs. And because it's pairs, we think double. That's why it's called a diploid cell. Now, let's look at meiosis. Meiosis is just about gamete production, making eggs, making sperm. Okay, and so for that reason, because the egg will be fertilized by the sperm, it only contains half the genetic information, not in pairs, just half the genetic information. Half is like the beginning of haploid, so they are haploid cells. Okay, and it's only about making gametes. Now, we had 1 division in meiosis, and we've got 2 divisions in meiosis. Okay, 1 division in mitosis, 2 divisions in meiosis. Now, the cells in mitosis were identical because it's about growth and repair. The cells in meiosis are non-identical. We get a jumbling up of the chromosomes, and that's really important because if that didn't happen and we always had exactly the same genetic information in all egg and sperm, then you would look exactly the same as brothers and sisters, even if you are not identical twins. Okay, so we must have variation in order to get the variation in the offspring. Okay, let's have a look at this, just meiosis as a picture. So in mitosis we had, and this is done in more detail on the mitosis video and the meiosis video that you can see separately, but in mitosis these chromosomes at this point split at the centromere. So we've got 2 chromatids joined together by a centromere. And in mitosis, they broke at the centromere, and 1 chromatid went to one cell and 1 went to the other. And that's what made them identical. Now, this doesn't happen here. So if we look, we've got the chromosomes copied, just like we did in mitosis. Okay, so we're looking at meiosis. Chromosomes copied just like mitosis. Then instead of splitting at the centromere and 1 chromatid going to each side, we actually see one of the chromosomes. Here we've got the large green chromosome has gone to this cell, and a small pink one. And then over here, from that pair of chromosomes, we've got the pink large one gone to this one, and the small green one gone to this one. So already on our first division, they are non-identical. Okay, on the second division, here's the second level of division. Okay, we've got those chromosomes splitting at the centromere, which is the bit that joins the two chromatids together. And we're left now with, these are not chromosomes, these are chromatids. Okay, so they've split at that centromere and they've become chromatids. And we can see there's less genetic information in these cells at this bottom level than there are in these cells. So we can see that they're non-identical because we can see there's variation by the different colours of the chromatids. And we can see that they have less genetic information than the original cell, because we've now produced these haploid cells that are then going to be, the egg will be fertilised by the sperm, giving it the pairs of chromosomes, getting it back up to that total, back up to a diploid cell. In a simple world, maybe if we're on foundation paper and we find that all a bit tricky, just think of meiosis as an extra stage after mitosis. So just think of it as we have these two cells and then they break again, splitting the genetic information to get 4 non-identical cells. But if we're on the higher paper or in triple science, we need to know that it's not, we've got the copying of the genetic information, but it's not splitting at the centromere in that first division. It's just one chromosome going to each, and then we split at the centromere and the chromatids go into these final cells. So meiosis, let's just do a quick recap. Meiosis, making 4 non-identical cells using 2 divisions. Okay, let's just have a little look at the introduction to DNA and chromosomes. So when we delve into the cell, we get into the nucleus. Within the nucleus, we've got chromosomes. If we're looking at a human, we say there are 23 pairs. It may be that the question is about a different animal or a plant, and there are a different number of chromosomes. Don't be put off, but just use the rules you know. DNA is a polymer. It is a double helix. Sometimes that's a multiple choice question. So it's a polymer and it's a double helix. Okay, we look at one of those chromosomes, we can unravel it, and we see that a section of chromosome is called a gene. Okay, and in here we can see these, like rungs on a ladder, we've got these base pairs. Okay, and when you study the triple science, you need to know more about those. So look at the booster video for that up to triple science. So we've got these sections of base pairs. Now 3 base pairs together is 1 amino acid. Put lots of amino acids together, we've got a protein. So this is all protein. Okay, DNA, protein. And we've got a gene here, and a gene codes for a particular trait. Okay. Now, at some point in time, a few years ago now, the Human Genome Project was set up, and it was an international publicly funded project. Okay, and what they were trying to do, it was lots of scientists bringing together their projects to work out all of the possible human genes. Okay, they sequenced 3 billion bases. Okay, now bear in mind there's only 4 different bases. To get 3 billion combinations was really quite a feat. Okay, and they worked out there were between 20,000 and 25,000 different genes of the human genome. And when we say all of your DNA, your genes, we can refer to that as your genome. Okay, now why did they do this? They did this to learn about different types of disease that are genetically inherited. They did this to find out how they can understand and treat different inherited disorders, and they did it also to trace back migration patterns of humans in the past. Then we go on to look at genetic inheritance. Lots and lots of keywords here. Okay, we need to be able to understand the keywords so that we understand what they're asking of us in the question, and we need to be able to use the keywords in our answers. So we've got the chromosome. Section of chromosome is the gene. It codes for a particular trait or characteristic. Then we have an allele. An allele is probably the most difficult word for people to remember. It's the different version of the gene. So it could be that the gene is for eye colour, and it could be that the allele is brown or blue. It's the version of that particular trait. The genotype is any two alleles together. So we've got big B, big B, big B, little b, or two little b's, and we would describe any of those as the genotype. Okay, the combination of genes. Then we've got phenotype. Think of the first two letters, PH. It's the physical expression. What do we actually see the person to have? For example, blue eyes. Then we've got the word homozygous. Homozygous meaning the same. This could be two big letter alleles together or two small letter alleles together. We could describe someone as being homozygous dominant for a particular trait, in which case they would have two large letter alleles. We could describe them as being homozygous recessive. They would have two lowercase alleles. Heterozygous, meaning one of each. So here we've got big H, little h. So again, heterozygous genotype. And then dominant, now this is an interesting definition that sometimes crops up and it's a hard one to explain. Okay, a dominant trait is something that will always be expressed even if there is only one of these alleles present. In other words, you will have a dominant trait expressed if you have a homozygous dominant genotype, for example, big B, or if you have a heterozygous genotype, for example, big B, little b, because that dominant allele will always express the dominant feature when only one is present. Recessive, both alleles need to be present as recessive alleles, lowercase letters, in order to have that trait. 2 inherited disorders. The likelihood of us seeing these in the exam is slim. There might be a totally different inherited disorder that they talk about, but so long as you know, is it dominant, is it recessive, you'll be able to answer the questions. You use the same ideas for each. Polydactyly, having an extra finger or toe, not a life-changing situation to be in, so you're probably not going to have embryo screening to find out if your offspring's going to have it. Because it's a dominant trait, it's actually quite common. And because it's a dominant trait, it will be expressed even if only one of those alleles is dominant. So as we can see there, it says it's caused by 2 big dominant, big B's, so dominant alleles, or heterozygous genotype, 1 big letter, 1 small letter, because that dominant trait only needs to be there present in 1 allele in order to have that trait. And we've done a Punnett square here. In any Punnett square, make sure you analyse the Punnett square. So we also need to add to this which one is mum and which one is dad. So you can add a little label here and write mum, and this is dad here. And it represents mum's potential eggs. Okay, she carries this allele and this allele, and dad's potential sperm, this allele and this allele. And then we match them up and we work out our analysis. And we've got here circled, we've got a 50% chance that the offspring will have polydactyly. Cystic fibrosis is caused when lots of mucus builds up around the major body organs. It can be a problem for breathing because it stops that diffusion of gases going from the alveoli to the blood, and it can be a problem for the digestive system because it reduces the amount of absorption of nutrients from the small intestine into the blood. It's a recessive trait. It can only be caused by having 2 recessive alleles, little b, little b. That's homozygous recessive. If somebody has a heterozygous genotype, big B, little b, they carry the potential to pass that condition onto their offspring, but they don't have it themselves. They're described as a carrier. So when we analyse one of these Punnett squares, we need to put a bit more detail into it, okay? Remember as well, that's not on here, mum, dad. It could be either way around. You could put mum here and dad here, but do what the question tells you to do, you know, label them correctly. So we're saying here, well, what happens if we have two carriers? Okay, we've got 25% here will not have cystic fibrosis. We've got 50% here will have one of those cystic fibrosis alleles, which we know means that they are a carrier. And then we've got 25% down here will have cystic fibrosis. So if two people who are both carriers of cystic fibrosis have a child, the chance of them having cystic fibrosis is 25%. The chance of those offspring being a carrier themselves, 50%. Now, we might want to have embryo screening. Embryo screening is where we take some cells, or the doctor takes some cells from the unborn child, from the embryo, and they are tested to see if they have a particular genetic disorder. The couple may want to do this if they think that there is a chance that the child will have a particular genetic disease or disorder that if they find out they have it, they might not want to have that baby. So it's not really a good idea to have embryo screening if you think to yourself, well, actually, it doesn't matter what the results bring me, I'm going to have this child and love this child, okay? But if you're thinking, well, actually, I think if they have, for example, cystic fibrosis, maybe I don't want a child to have to go through that, then you may consider the options for termination. There's lots and lots of ethical issues here, okay? Even doing embryo screening, there is a possible miscarriage, and people refer to this process as designer babies, this idea of thinking, okay, well, I'll have a baby if it's got these features because these are the features I'm looking for. Okay, one to consider. Sex determination, okay, last use of a Punnett square. So and this is going to give us some quick, easy marks, okay. The chromosomes, the sex chromosomes in females are XX, in males XY. Okay, so that could be a really quick 1 mark question. Okay, what's the sex chromosomes in females? XX, in males XY. So here is the possibilities for the male sperm. So we could write dad here, and here are the possibilities for the female eggs. We could write mum here. We match them together in our same way. So match, 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 and match. And we see that there is a clear 50% chance that the offspring will be a boy and a 50% chance that the offspring will be a girl. And that makes sense because roughly on Earth, there are the same number of men and women, or males and females. Okay, variation. In sexual reproduction, offspring receive some characteristics from each parent. We said that. We said that sexual reproduction is good for offering variation, genetic information, chromosomes coming from both parents. However, there's also variants arise from mutation, and mutations can be so minor. It's like when you make a spelling mistake and you can still work out what the word is. They make no difference at all. However, sometimes if the mutation is significant enough, it can create a new feature. Now, if that feature is beneficial, then we start going into what Darwin talked about with natural selection. That individual will live longer, it will reproduce and pass on that mutation. If the mutation is not useful, then the individual will probably, and we're thinking about in the animal world here, will probably die out before it has a chance to pass it on, in which case it doesn't pass on to future offspring. Now, these mutations occur all the time, okay? If it's suited, we'll see those changes, okay? We'll see that evolution over time. If it's not useful, it will just fade away. Now, scientists believe that evolution is the change in inherited characteristics of a population over time. Remember, we said a population is of the same species, and this is through a process of natural selection. The theory of evolution by natural selection states that all species of living things have evolved from simple life forms that first developed more than 3 billion years ago. And the theory of evolution by natural selection, referred to as survival of the fittest, is what Darwin talked about. Now, this is quite often a 3 or 4 mark question. If there has been a mutation that is beneficial, for example, a longer neck to reach food higher up, then the animal will be a better competitor. Okay, 1 mark. Live longer to reproduce, 2nd mark, potentially another two marks there. And then it will pass on that gene to its offspring. Okay, and so this will continue over time. And we called that natural selection, survival of the fittest. The fittest get to survive to pass on to their offspring. And as I said before, if the mutation is not beneficial, the organism will die and the mutation will not be passed on. So, if the feature is good, the organism, could be plant, could be animal, will be a better competitor. It will live longer, therefore have more time to reproduce and pass on that gene to its offspring. Now, we've got lots of things here. Okay, if they have 2 species have become so different that when they interbreed, they produce an infertile offspring, we can categorically say that those 2 individuals are different species. The example we looked at was the zedonks at Colchester Zoo. Okay, so one night a donkey got in with the zebras and they had 4 zedonk offspring. Now, because the zebra and the donkey are different species, the zedonks that were created, that were born there, are infertile. And that's how we can categorically say donkeys and zebras are separate species. Because if 2 organisms mate together, reproduce and produce fertile offspring, we say they're the same species. Infertile offspring, not the same species. And fertility is about, can they have offspring of their own? So the zedonk cannot have offspring of its own. Therefore, we say it's infertile. And we can see this, which is called an evolutionary tree, comes up quite often as a 1 mark answer. Here we have a timescale. This is present day, and this is 3.9 million years ago. And we can see lots of different species, and we can see the time at which they became separate species. So the 2 species here that are most similar are these 2 at the top here, because they're on the shortest branch. Okay, this one at the bottom became different from the others 3.9 million years ago. Okay, why they've done this one with a pointed, I think it's just showing you a different way of drawing it. Sometimes this whole evolutionary tree is picked up and turned around so you see the species at the top. Either way, we can use this time scale here to tell when did these 2 become separate species. We could follow it down, read it off this scale at the bottom. Okay, so it's showing us when they have become separate species. Let's look at selective breeding. So natural selection happens in nature, we're not part of it. Selective breeding has been happening for thousands of years. It's a very long process involving human intervention. We have selected the individuals we would like to breed together. So a farmer looks at all of the animals that they've got and they decide, I really like the characteristics of that animal, I really like the characteristics of that animal. If I breed those 2 together, I may get offspring that have exactly the characteristics, the traits that I want. And we can see how the chicken has changed over time. We don't suddenly get to this huge chicken straight away. It has to be repeated over many generations. Now that's really important. Okay, so natural selection happens naturally in nature, no human intervention. Selective breeding, we are involved. This isn't genetic modification. This is just selecting the animals we want or the plants we want to breed together to try and get kind of the ultimate offspring, the one with the features that we really want. Okay, and it happens over many generations until we get the desired characteristic. And we've been doing this for thousands of years. There was a mutation in a cow. Okay, these are what cows should look like. They don't look like that anymore because we've been selectively breeding them to have more meat. But there was a mutation in a cow that meant that the muscles kept on growing and growing. And they thought, this is brilliant. We'll get loads of meat from these cows. And so they selectively bred the cow that had the mutation with a big bull. and then they kept selecting the most muscly cows and bulls to breed together until they got this variety that they call the Belgian blue, which is really muscly, lots and lots of meat there. Okay, for triple science, it was the switch part of the gene which had broken, which had mutated, which caused the growth to not stop, basically. Okay, so if you're doing triple science, just that little add in there. Okay, you've got the coding part and the non-coding part. The bit which represents the switch, there was a mutation there, and so it didn't switch off growth. Okay, so it just kept on growing and growing. Okay, why would we want to use selective breeding? To get crops that are resistant to disease or resistant to frost, to produce animals that have produce more materials for us in terms of meat or milk, for domestic dogs with a gentle nature, or for large or unusual flowers. Okay, with this process of selective breeding, we can get inbreeding. Okay, and that means that you get breeds that are prone to disease or inherited defects, and that can happen where a small population of individuals is used. It's gone to genetic engineering. So we've done natural selection, we've done selective breeding, we're now on to genetic modification. This is where we're actually changing the genetic makeup. So we are not removing a gene, we are only adding a gene. We're adding one gene, one trait, one characteristic to an existing set of chromosomes. Okay, why would we want to do this? Okay, if it's plants, we could want to get pest resistance, frost resistance, disease resistance, herbicide resistance, so we can spray all of the crops and the weeds die but the crop doesn't. Drought resistance or longer shelf life, and this is really, really useful in getting higher yield and more profit. Let's have a look at the process of genetic modification in order to create insulin for diabetics. Now this links into B5. So here we have a cell from a person that is able to produce insulin controlling blood sugar levels. And what they do is they find and isolate the gene that makes the insulin making gene, and it's cut out using enzymes. Now we know that bacteria can multiply really quickly. So if we take a bacterium, take out some of its genetic information in the form of one of these plasmids, again that's opened up using enzymes, and that specific gene is added to the plasmid. The plasmid is returned to the bacterium and it multiplies rapidly, so we end up with a whole group of bacteria that all have the ability to make insulin. The bacteria make lots and lots of insulin, and we use that in our diabetics, the pens for type 1 diabetes. Really, really good use of genetic modification. Advantages of genetic modification: we have fewer chemical sprays needed, bigger yields, which I said already, they can grow in harsher conditions, and overall food will be cheaper. We've got the disadvantages of if there's an organic crop next door, then they may well be contaminated. Once it's out there, it's hard to get it, to bring it all back. We don't know the implications of genetically modified food. It could reduce biodiversity, and also people can be allergic to it. Okay, it can be expensive as well, because obviously it's a desirable thing to have some crops that are potentially going to give a bigger yield and more profit. Okay, let's have a look at fossils. 4 ways of creating fossils. Okay, we've got mineralization. This is our typical fossil. Animal dies, lands in sediment. Sediments lands on top of it and it becomes mineralized. It leaves an impression and minerals fill in that gap. Okay, we've got footprints in mud that then solidify, so leaving a print of the animal that's walked through it. And then we've got our 2 methods that preserve the entire organism. This is tree sap. Okay, animal lands or small insect lands in the tree sap. It then gets covered with it. and it turns into amber, or we could have some kind of avalanche or ice age where animals get trapped in the ice and it preserves the whole organism. Okay, so both of these preserve the whole organism. Now we can age fossils using carbon dating. Sometimes we see that there are gaps in the fossil record, and this could be that fossils haven't been discovered yet because they're in extreme places under the ground, places where people don't go, or it could be that they've been destroyed by natural disasters, earthquakes for example, or building work. Some organisms we don't know much about at all. These are the soft-bodied organisms. These are ones that decay before fossils can form. It does not account for gaps in the fossil record, but it tells us we have no knowledge of these particular organisms. So next we'll look at extinction. Now we cover this again in B7 and we looked at it as a 6-mark question. What factors cause extinction? Now it could be one of 6 factors: disease, new pathogen to the area, loss of habitat, loss of food, a new predator, or that could include hunting by humans, climate change, or natural disaster, which encompasses all of the different things like earthquake, volcanic eruption, tsunami, all of those. Okay, and then what could the population do to survive? Well, they could separate their communities so they don't all stick together, gives greater chance of survival. They could find a new territory, they could change their food source, adapt, or migrate to a new area. Now, the next part is on resistant bacteria, and this came up in paper 1, and I've actually just left it as the course content because it's everything on here we have covered before. So we know that bacteria rapidly reproduce by that process called binary fission. We know that sometimes the bacteria can mutate, creating a new strain of bacteria that will be resistant to antibiotics, and the ones that survive and aren't killed by the antibiotics reproduce, creating more of those resistant strains, which links to that idea of natural selection. Okay, so if we've got this big population that can be killed by the antibiotics and then a small mutated population, if we overuse those antibiotics and we prescribe them regularly by the doctor, then it means that the mutated strain actually have less competition and therefore will reproduce a lot faster. So we don't want to overuse our antibiotics. We also said that we shouldn't inappropriately use antibiotics. This is where we've been given antibiotics by the doctor for 7 days and we take them for 4 days, we feel much better, so we stop taking the antibiotics. Now there is a low level of bacteria inside the body which we can have there and we feel totally fine. So we might feel okay and think that the antibiotics have done their job, but we've still got that low level of bacteria in the body. As soon as we stop taking the antibiotics, those bacteria are going to start multiplying by that binary fission again, and then we will feel ill again. So we must take the whole course of antibiotics. Then we've got things like MRSA, which is a massive problem in hospitals, very contagious, and it's resistant to antibiotics. It mutates so readily that they can't keep up with those mutations by creating the right antibiotics. Okay, last slide. Classification of living organisms. There were 2 guys, both called Carl Linnaeus and Carl Woese. And Carl Linnaeus came up with the Linnaean system of classifying living organisms. This is the one we're familiar with, the one where we start off with all living things, and then we've got our animals and our plants, and we go into the animals group, and we've got our vertebrates and our invertebrates, and so on. What we need to remember for the Linnaean system is King Philip came out for Great Soup, and every level as it branches more and more specifically has a level. Okay, so we start off with the kingdom, that could be the plants or the animals. We go down to the phylum, that could be the vertebrates, the invertebrates. We go down to the class, that could be within the vertebrate group, that could be the birds, the reptiles, the mammals, the fish or the amphibians, and so on and so forth. We've then got our final genus and species. Now we can see here the first word is the genus and the second word is the species, and we can see that cat and tiger have the same genus, so they are similarly related because they have the same genus, but we know that they are separate species because we can see catus and tigris. Okay, this is known as binomial classification. Bi because there's two names to it. Okay, so the genus is the first name and the species is the final name. Then Carl Woese came along. Carl Woese only died in 2012, and he said, looking genetics only, we can actually classify, and the structure of these living things, we can classify them into 3 domains, and he named it the 3-domain system. Here are the 3 domains. Now all we need to know is the names of these 3 domains. We've got the bacteria, the true bacteria. We've got the archaea. These are the primitive bacteria. These can live in extreme conditions such as those hydrothermal vents which are deep under the sea. And then we've got the eukaryota, and these include the protists, the fungi, the plants and the animals. Plants and animals, separate kingdoms in this system. In this system, they're in the same domain. We need to be aware of both of the names of these 2 Carls and also the systems that they created. Hope you found that useful. Thank you.